Good evening, everyone. Good afternoon to those of you that are joining us from the West Coast. My name is Nicola Worsfold, and I'm the Executive Director at Defeat Duchenne Canada and a Duchenne mom. Defeat Duchenne Canada is a national organization representing individuals and families living with Duchenne muscular dystrophy across our country. Our work is grounded, can you go to the next slide, please? Our work is grounded in four key strategic pillars. We are the nation's leading funder of Duchenne research. We support families by advocating for access to the best treatments and care, and by delivering first-in-class educational programs like this one this evening, which will be our final webinar of the year. It is my pleasure to welcome Avidity Biosciences this evening to share their updates on their RNA therapies and clinical trials in Duchenne. We were very fortunate to have Avidity join our family forum earlier this year, and we are grateful to have them join us again this evening. So tonight we are joined by Beth Hendrickson, Director of Patient Advocacy, and Elizabeth Ackermann, Senior Vice President and Head of Clinical Development. Welcome, Beth and Elizabeth. Beth, I'll turn it over to you now.
Thank you, Nicola. I'll share my screen here for you all. Okay, well, hello and welcome. My name is Beth Hendrickson, and I am part of the advocacy team at Avidity Biosciences. And we're pleased to have you join us today as we review the del-zota Explore 44 OLE top-line and functional data. Before we get started, we'd like to thank each study participant, so the boys and men living with Duchenne, amenable to exon 44 skipping, their families, investigators, and their teams for the time, commitment, and continued contributions to the studies. Thank you. I'm going to cover a couple of upfront slides, and then we'll get started in our formal presentation. A few disclaimers to cover for you, as the presentation today is at the request of Defeat Duchenne Canada, and the presentation discusses a clinical development program for an investigational product, meaning that the product has not yet been approved by the FDA or Health Canada. So therefore, conclusions regarding the efficacy or safety cannot be drawn from this data. So the data is being shared for educational purposes and therefore should not be used to make individual treatment decisions. The data presented today is from a phase 1 and 2 study, which is evaluating the safety and dosing of the product we refer to as del-zota. So phase 2 of Explore 44 is an open label extension study evaluating the safety and tolerability of long-term dosing of del-zota. So once again, del-zota is an investigational product and not approved by regulatory agencies. Its safety and efficacy have not been established, and Health Canada has not granted market authorization. What can you expect to hear from us today? We'll cover the 4 topics on the left-hand side of your screen, starting with an introduction to Avidity, and cover a brief overview of our science. Then we'll talk about exon skipping, both as it relates to Duchenne and then specifically to del-zota. And then we'll cover our data, which is why we're all gathered here today. And we'll make sure there's a time at the end for a few question and answers. Today, we're joined by Lisa Ackermann, who is the Head of Clinical Development and Senior Vice President at Avidity. Lisa has been with Avidity for a number of years and for the industry for over 20 years, so she's certainly well-versed in the data you'll be presented with today. With that, I'll turn it over to Lisa to get started and introduce Avidity.
Thanks, Beth. It's really an honor and a privilege to be here today. So I've heard from many of you in the community over the years how difficult it was to hear the diagnosis of Duchenne's for the first time. And I know that this diagnosis brings many of you on a journey to find ways to help support your loved ones, such as getting involved in advocacy groups, or maybe looking for opportunities in clinical research, or investigating what kind of therapeutics might be available to you. So today I'm going to talk about a potential drug that we are developing for DMD amenable to exon 44 skipping. And as Beth said, we'll be explaining how our technology works and how we're applying it to DMD. It is our hope that this information that we are sharing with you today might be useful to you and your family on your journey. So at Avidity, our vision is to profoundly improve people's lives by revolutionizing the delivery of RNA therapeutics. Now, you might be asking, what are RNA therapeutics? They're actually just a very specific type of drug that works by binding to RNA, which is found in every cell in your body. I'll also be referring to them as oligonucleotides or oligos. So oligos are really powerful types of drugs because they have the ability to target the underlying cause, genetic cause of a disease. At Avidity, our primary focus has been to develop these drugs for neuromuscular diseases such as DMD. So oligos have actually been around for long time now, but their use in neuromuscular diseases has been limited by really one key problem, and that is that they just don't get into muscle and cardiac cells very well. So we've developed a novel way to deliver these types of drugs into both muscle and heart cells. So this slide shows kind of the basics of how our system works. So we have developed a platform that we call antibody oligonucleotide conjugates, or AOC for short. And AOC is basically made up of two parts that we show here. One is an antibody and the other is an oligo. And they each have two very different jobs. The antibody's job is simply to deliver the oligo into the muscle and the heart cell. And on the next slide, I'm going to show you how that actually works. And the oligo's job is simply to bind to a very specific target RNA inside of the cell. In the case of del-zota, the oligo binds to the dystrophin RNA and causes the cell to skip over exon 44. And that's really shown in this cartoon right here. So you can see the AOC, or the antibody attached to the oligos, on the left side of this figure. So you can kind of imagine it circulating around in the bloodstream. And when it sees a muscle cell, the antibody part of the AOC binds to a receptor that is sitting on the outside of that muscle cell. After it binds, the receptor pulls the AOC into the cell. Once in the cell, the oligo is released from the antibody and it moves to the nucleus where it can do its job. And like I said, del-zota, the job of this oligo is to bind to the dystrophin RNA and cause the cell to skip over exon 44 so that the cell can now make dystrophin protein. So we wanted to talk to you a little bit more about this idea of exon skipping and how it works. So as you probably know, dystrophin is the largest protein in the human body. DMD is caused by mistakes or mutations in the dystrophin gene, which prevent the body from making dystrophin protein. Because dystrophin is so large, the dystrophin RNA, which is basically a recipe that tells the cell how to make dystrophin protein, this dystrophin RNA has 79 different exons, which we're showing here. Now, all 79 of these exons need to be lined up perfectly, like a puzzle, in order for this recipe to work and for the cell to be able to make the dystrophin protein. If pieces of the puzzle don't line up just perfectly or are incomplete, maybe due to a mutation in DMD, then the cell can't make the dystrophin protein. And we show an example of that on the next slide. So in this example, you can see that we're missing exon 44. And what you have left is we're missing exon 45. And what you have left is exon 44 and exon 46, which just don't fit together right. And so the recipe is broken and very little dystrophin can be made. But some smart person a while ago figured out in this particular case, if you cut out exon 44, and what you have left is exon 43 connecting to exon 46 that fit perfectly together, they restore the recipe and they allow the cell to make a shorter but functional dystrophin protein. And this is really exactly what del-zota was designed to do. It was designed to cause the cell to cut out exon 44 from the dystrophin RNA and to allow the cell to make the dystrophin protein. However, this approach only works if the dystrophin mutation that is present is amenable to exon 44 skipping. So what do we mean by exon 44 skipping? It's just that there is a particular mutation that if you skip over exon 44, it will fix the puzzle and allow the body to produce the dystrophin protein. Now, there are about 46 different dystrophin mutations or deletions that we know of that are amenable to exon 44 skipping, and we're showing those mutations here. So it's really important that you know your mutation so you can understand what exon skipping therapies might work for you. So that's sort of how the drug works. And now I'm going to go over the clinical trials that we've conducted with del-zota. So we've conducted two trials. The first one was called Explore 44, and it's now complete. And the second one was called Explore 44 OLE, and this trial is still ongoing. Explore 44 was a first-in-human phase 1/2 study. It was randomized, double-blind, placebo-controlled, which means that some of the participants got placebo and some of them had del-zota. And we looked at two different dose levels in the study, 5 and 10 mg per kg. The drug is delivered by IV infusions every 6 or 8 weeks for 3 doses. So in this study, we took biopsies at the beginning of the study and then again at the end of the study after the 3 doses. Participants that completed the Explore 44 can enroll into the Explore 44 OLE. And in the Explore 44 OLE, everybody received del-zota, and all participants are now receiving it at our target selected dose, which is 5 mg per kg. The Explore OLE enrolled 39 participants. And if you can go forward, there we go. There's the characteristics. They were age 7 to 27. About 2/3 of them were ambulatory and about 1/3 was non-ambulatory. And the functional data that we're going to show you today are from the participants in the Explore 44 OLE trial that have been on del-zota for about 1 year. So in the next slide, we have the safety data from the Explore 44 OLE. About 85% of participants had at least one adverse event, and 26% had at least one AE that was thought to be related to del-zota. There were 3 serious AEs, and one of these, hypersensitivity, was thought to be related to del-zota, and this serious AE led to discontinuation from the study drug. If you go to the next slide. So now I'm going to show the data from those biopsies that we collected at the beginning, at the end of the study, so after the 3 doses. And in those biopsies, we measured the amount of dystrophin protein, and that's what's shown here. So at the far left side of this graph, you can see the placebo group, and there's no change in their dystrophin expression before or after treatment. But in both the 5 and the 10 mg per kg groups, we saw an average increase of about 25% of normal dystrophin. And then another way to look at dystrophin, we're showing on the right, you can look at the distribution of the dystrophin that is made in the muscle cells through imaging. So this top image is a baseline image, so before the participant received del-zota, and you can see a few of the muscle fibers lighting up in red. These are called revertant fibers. But then if you then take an image after treatment, which is showed below, you can see a very different pattern here, and many more of the muscle cells are showing dystrophin expression, and they're showing expression in the correct location. So on the next slide, this shows the creatinine kinase levels that we measured after treatment with del-zota. So when the muscle cells are damaged or become fragile, the creatinine kinase inside the muscle is leaked out into the bloodstream, and we're able to measure it. So the blue circles of this graph represent individuals that received del-zota treatment in Explore 44, and we saw a very rapid decrease in their serum creatinine levels that was maintained to near normal levels for over 1 year of treatment. The gray triangles that you see on this graph are individuals that had placebo in Explore 44. So you can see when they were receiving placebo, there was really no change in their creatinine kinase. But as soon as they entered the open label extension study, which is the shaded area on the right, you can see that their creatinine kinase levels did decrease down again to near normal levels. So overall, we saw serum creatinine kinase decreases was reduced by greater than 80% from baseline. And at 1 year, about half of the participants had creatinine kinase levels that were in the normal range. So now we're going to show you some functional data. And what we're going to do is we're going to compare this functional data from participants who were treated for greater than a year with del-zota, and we're going to compare it to some individuals that participated in a natural history study. So the natural history study that we're using was done a while ago, and this data is now being held by Cure Duchenne. In this study, there were actually 269 DMD individuals. And what we did was we just picked out those individuals who had mutations that were amenable to exon 44 and had baseline characteristics that were similar to the del-zota study participants that we want to compare them to. So this left us with 22 individuals in this natural history comparison. So on the table on the right, what we're doing here is we're comparing the baseline functional status of the two groups. And if you can just go forward one more, thanks, Beth. You can see in the natural history group that the average age was 10.6, and in the del-zota group, the average age was 11. But then if you look at each one of those time functional tests, the 4-stair climb, the 10-meter walk run, and the time to rise from the floor, you can see that the del-zota group took longer to complete each one of these tests. So that actually indicates that they were more severely affected than our control group. So before we start to show you some of the data comparing the del-zota group with the natural history group, we wanted to talk a little bit about the tests that we're looking at, and then how you can kind of look at these graphs. So time functional tests are meant to measure the impact of Duchenne on motor functions, such as climbing stairs or getting up from the floor. And when we do these tests, we're simply just capturing how long it takes to complete the task. So on the right-hand side of the slide, you can see that if a task takes longer to complete, that the change from baseline will go to the left, and this means that there is a worsening. But if an individual completes that task in a shorter period of time, the change from baseline will go to the right, and this indicates an improvement. So we give that a little thumbs up. All right, so on the next slide, what we're seeing here is a snapshot of the data from the natural history study that I mentioned before. And so this shows the change from that study over a 1-year time frame for each one of these different time functional tests, and also the poll, which is at the bottom. So you can see in the natural history cohort that they really took longer to complete these tasks by 1 or 2 seconds over that 1-year time frame. So there was a worsening in all of these tests over that 1 year, and also in the poll, which is at the bottom, and that is a test for upper limb mobility and function. So if you go to the next slide, so now we're going to take these one by one. So this is the 4-stair climb, and we're comparing it now with the natural history data, with comparing it to the del-zota group that have been treated for about 1 year. So you can see again that the natural history participants took longer to do the 4-stair climb, while the del-zota group on the right, actually after 1 year time frame, there took almost 2 seconds less to complete this task. So that did show improvement. On the next slide, we have the 10-meter walk run test. And this is a test where a participant is asked to run or walk over a short distance. You can see again that the del-zota group took less time to complete this test over that 1 year, while the natural history group worsened and they took longer to complete that test. So again, we're showing the thumbs up as a good thing. And then finally, for the time functional test, we have the time to rise from the floor. Again, the same sort of pattern. You can see that the del-zota group improved, took less time to do the test, while the natural history cohort actually worsened over that 1 year time period. So another test besides the time functional test that is often used is called the North Star Ambulatory Assessment. And this is an assessment that looks at skills such as raising your arms, standing on one foot, or maybe hopping on a foot. And this test has a score that ranges from 1 to 34. So a higher number on this test is better. So as you can see in the graph, if the number goes up, it's an improvement. And if the number goes down, it's worsening, sort of very similar to the time functional tests. So on the next slide is the data. Again, you can see that the natural history group worsened by 2 points in this test, while the del-zota group was stable, that basically didn't show any change over that 1 year time frame. And then finally, the last data slide we're going to show is with the performance of upper limb or the pull test. Now, this is an important test because it can be performed on both ambulatory and non-ambulatory individuals. Those tests that I was showing you previously can only be done on ambulatory individuals. So again, we're comparing a natural history. Now, actually, this is data that was published in the literature. Again, only DMD 44 individuals over a 1 year time frame, and they got worse on the pull, while our del-zota group, after being treated for 1 year, improved by 1.5 points on the pull scale. So very consistent with the time functional test and the NSAA. So in summary, in this exploratory analysis of del-zota showed improvements across a number of key functional endpoints after 1 year of treatment. We also saw significant increases in dystrophin protein and a sustained and significant decrease in creatinine kinase. Del-zota also had a supportive long-term safety and tolerability profile. So we are really excited about this exploratory data that is emerging from the open label extension trial and believe that the changes that we saw in dystrophin in that first study, Explore 44, are translating into early signs of potential functional benefit. And this supports a continued development of this therapy. So what's next? So our team is very busy planning to submit these studies to the FDA in a biologic license application in 2026. And we hope that this application will support an accelerated approval of del-zota. We are also in the planning stages of a confirmatory phase 3 trial. This will be a global program, and we look forward to sharing more information with you about this program when it is available. And finally, we are also actively working on another program for DMD, which is for individuals that have mutations amenable to exon 45 skipping. So that's it. I'd like to close to again thank the individuals, the families, and the sites that participated in Explore 44 and Explore 44 OLE, and also to those participants who participated in the natural history study. So your involvement, the time, and the commitment has been critical to supporting the advancement of new therapies for DMD. So thank you.
Thank you, Lisa. That was really good. That was a great presentation. You did a really great job of explaining how exon skipping works because it's never an easy one to communicate, and you did an excellent job. That was very clear. Thank you for that. So, does anyone in the audience have any questions? If you do, feel free to type your questions in the Q&A box. So there should be a Q&A tab at the bottom of your Zoom call that you can click on and type in your questions, and we'll be happy to answer any questions. In the meantime, while we're waiting for questions to come in, I have a couple of questions myself, and you mentioned that you're going to be starting a phase 3 global program very soon, which is really exciting. Is there opportunities to potentially have trials in Canada?
Yeah, so we're right now in the planning stages of the trial, and we're talking to the regulatory authorities globally. And so, as soon as we have more information about exactly what regions this trial will be located at, we'll be sure to make that information available.
Amazing. That would be great. And we always encourage families to check in with your clinicians and ask about when trials are becoming available. And of course, we'll be happy to post any updates through our news channels as well to update families on when these types of trials will be available to Canadian patients. So that's really great. Thank you. So just looking at the Q&A, I think we have a question. So we have a question from Doyle, and he says, what about a multiple number of exon skips, i.e., for example, 10 numbers in a row?
So, so there are, I'm a little, if you're talking about there are mutations that have multiple exons, like 10 exons in a row that have been deleted. There are, for exon 44 amenable mutations, there are a number of mutations that have a large number of exons deleted in that mutation, more than 10 in a row, that the exon 44 amenable therapeutic would work for. And I'm not sure if that was exactly the question, but
I think so. And I think, as you said, it depends on what the numbers are, right?
yeah,
And this is why we always say to families, like, make sure you know your numbers, like know your mutation, because in instances like this for exon skipping, it does matter. And I think you had a slide up, Lisa. I don't know if you want to throw it back up again. The one that had the chart
yeah.
with all the possible exon skipping deletions that were amenable to 44.
Yeah, I think maybe if Beth can throw it up because I know, for example, on the top of my head, there's a deletion for 3 to 43 that is amenable to exon
Oh,
skipping.
there we go. Yeah.
Yeah, there it was.
Yeah.
But yeah, it's the first one listed. So that is a mutation that has a lot of the exons deleted in a row, and it is amenable to exon 44 skipping. So again, know your numbers, know your mutations, ask your doctor so you can understand which one of the exon th skipping therapies might work.
Amazing. Thank you. And Doyle, hopefully that answered your question. Oh, hang on. I think we have, oh, no. Okay. Any other questions from our audience? I know we're getting close to the end of our time. We will be posting this webinar live on our website. So if you did miss it, you will certainly have an opportunity to see it again. And I just want to say thank you to Lisa and Beth. We really appreciate you coming on and sharing this great news. Your program looks fantastic and looks like the results so far are looking very promising. So we really do look forward to your trials that are coming up and hearing continued updates on how the program is progressing, and particularly opening it up to individuals who are amenable to exon 45 as well. Will certainly be other opportunities for families. So thank you so much for joining us today. Thank you for coming. I'll pass it over to Will now just to close us out. But thank you everyone online for joining us this evening. Will, do you want to take it from here?
Thank you, Nicola. So just as a reminder, we would love you to join our conversations and to follow us on our social media platforms. You can see here all the platforms that we're on, and you can join us through at defeat Duchenne and hashtag defeat Duchenne. Next slide. Also, too, we would love you to sign up for our newsletter, and this is where you'll get lots of information about what's going on with the organization and within our community. So you can join the newsletter through defeatduchenne.ca. And next slide. And we would love to hear from you as well. We have an evaluation, and I believe Rafaela is going to post in the chat box. And we will also be sending it out to the folks that attended today, tomorrow. So you'll have time to be able to fill that out. We really appreciate your feedback, and it helps us to know what we did great and where we can improve for our future webinars. So thank you very much.